Hello friends, welcome to the student world. Today we will discuss about pBR322 as a cloning vector. Let's talk about what is pBR322. pBR322 is a plasmid vector which is constructed in lab by two scientists, Francisco Bolivar and R.L. Rodriguez, in the year 1977. Now what is the meaning of this word pBR322? The name denotes P stands for plasmid, B and R the name of the scientist who developed it in the lab, B for Bolivar and R for Rodriguez. 322 is the number which differentiate it from the other plasmids that were made in the same laboratory, for example pBR325 and pBR327. Now how we constructed this plasmid in the laboratory. In the construction of pBR322 vector, we have taken sequence from three natural plasmids: ampicillin resistant gene from plasmid RSF2124, tetracycline resistant gene from PSC101 plasmid, and the replicon. Replicon contain the origin of replication and all its control element. This replicon is taken from natural plasmid PMB1. In this way, pBR322 vector is constructed in the laboratory. Let's talk about characteristic feature of pBR322. First of all, size. The size of this vector is 4063 base pair. That is, the size of this vector is ideal for a good cloning vector. Second, it contains two set of antibiotic resistant gene, ampicillin resistant gene and tetracycline resistant gene. These genes are the marker genes that help in simple identification of the transformed cell. Transformed cell is a cell which contains recombinant vector. The entire genome of pBR322 contains 40 restriction sites, out of which 11 are present in tetracycline resistant gene, 6 within the ampicillin resistant gene, and 2 sites are present in the promoter region of tetracycline resistant gene. We can use any of these 19 restriction sites for the cloning. Now the vector contain an origin of replication and a gene rope which encode a protein that help in the regulation of number of copies of the plasmid that are present in the cell. This plasmid have copy number of 15 to 20 per cell. Copy number is the average number of plasmids present in a cell. This plasmid number can be increased by giving chloramphenicol treatment. Now you see the diagram of pBR322 vector. On the right hand side you see the tetracycline resistant gene, and on left hand side there is ampicillin resistant gene. At the bottom you see there is origin of replication. Origin of replication is the site at which replication of the plasmid start. And rope gene, it encode a protein which control the copy number of this plasmid. The restriction sites present in both resistant gene are used for cloning. For example, BamHi site in tetracycline resistant gene and PST1 restriction site in ampicillin resistant gene can be used for cloning. Now we will see how this rope protein control the replication of this plasmid. Above this is shown the origin of replication of this plasmid. This ORI region encode two RNA molecules, RNA1 and RNA2. RNA2, it act as a primer by binding at ORI region. After binding of this RNA second, RNase H enzyme trim this RNA to generate 3' OH group. Our DNA polymerase uses this 3' OH group and start replication. Now when the copy number of plasmid increases, RNA1 is also transcribed by another strand of DNA molecule. This RNA1 and RNA2, they are complementary in sequence. That's why they form a hybrid. When RNA1 and RNA2 hybrid is formed, RNA2 is not able to bind at ORI region and start replication. This hybrid of RNA1 and RNA2 is stabilized by this Rho protein. In this way, Rho protein control the replication of this plasmid. Now we will see how the low concentration of chloramphenicol increase the copy number of this plasmid. When we grow the bacterial cell having this plasmid in a medium containing the antibiotic chloramphenicol, this antibiotic inhibits the protein synthesis of the bacterial cell. When we use it in low concentration, the protein synthesis is inhibited partially. As we know, the initiation of bacterial chromosome replication require proteins, and when protein synthesis is inhibited, proteins are not available for initiation of bacterial chromosome replication. So the enzymes which are not used in chromosome replication are used in plasmid amplification. Plasmid replication doesn't require the protein for initiation of its replication. As we see earlier in the diagram, plasmid replication is initiated by an RNA molecule. When we give antibiotic treatment, the chromosomal replication stops, but plasmid replication continue. In this way, plasmid copy number increases when we give antibiotic treatment. We will see how we do cloning in this vector pBR322. First of all, we require a gene which we have to amplify. For this, we take a genomic DNA and digest this DNA with restriction enzyme BamHi. After digestion, we take a fragment which we have to clone in this vector. For cloning, we use tetracycline resistant gene of this vector. We cleave this vector with the same enzyme BamHi. After digestion, we insert this foreign DNA in tetracycline resistant gene and recombinant vector is formed. After recombinant vector formation, this recombinant vector is transferred in a bacterial cell. Now you see in the diagram, this is a recombinant vector and it is transferred in this bacterial cell. The process by which we transfer this recombinant vector in this bacterial cell is known as transformation. Now this bacterial cell is called transformed cell. After transformation, the characteristic of the vector are present in this bacterial cell. Now this vector contains two antibiotic resistant gene, ampicillin and tetracycline. This bacterial cell is ampicillin sensitive and tetracycline sensitive, and when this vector enter this cell, then this bacterial cell gain the ampicillin resistance. Now we grow this bacteria on a growth medium. This growth medium contain ampicillin antibiotic. This antibiotic is included in the medium because the first selection is whether the vector enter the bacterial cell or not. If the vector is present in the cell, then this bacterial cell is able to grow in this medium which contain the antibiotic ampicillin. If the bacterial cell doesn't have a vector, then this bacterial cell cannot grow on this growth medium. This bacterial cell gain resistance from the vector. Now how we will select transformed cell from non-transformed cell. The E.coli cell which has recombinant vector gain ampicillin resistance but not tetracycline resistance. We use replica plating technique for screening of these transformed E. coli cell. First we grow the bacterial cell with recombinant vector in a medium containing ampicillin, and then bacterial colonies are replica plated on another plate which contain the antibiotic tetracycline. Now the transformed E. coli cell is not able to grow on this tetracycline plate because the tetracycline resistant gene is inactivated by the insertion of insert DNA in it. Now you see in second plate few colonies are missing that were present in first plate. These colonies are colonies of transformed E. coli cells. Now what is insertional inactivation method which we use for the selection of the transformed cell? When a foreign DNA is inserted in a gene, then that gene becomes inactive and no protein is encoded by that gene. Now you see in the diagram, this is non-recombinant vector. It contains two antibiotic resistant genes and both these genes are functional. When we insert foreign DNA in this vector, this tetracycline resistant gene become inactive and ampicillin gene is intact. This recombinant vector now have ampicillin resistance but no tetracycline resistance, whereas the non-recombinant vector have both resistance, ampicillin resistance and tetracycline resistance. We use this insertional inactivation method for the selection of the transformed cell because if the transformed cell contain recombinant vector, then that cell have ampicillin resistance but no tetracycline resistance, and if this E. coli cell contain non-recombinant vector, then that E. coli has both resistance, ampicillin resistance and tetracycline resistance. Insertional inactivation is the inactivation of the gene by insertion of foreign DNA in that gene. After inactivation, that gene doesn't encode for any protein. Similarly, we can clone in ampicillin gene of pBR322. As we have cloned earlier, in the similar way, we can clone in this gene. First of all, genomic DNA and vector DNA is cleaved with PST1 restriction enzyme. After cleavage, insert DNA is ligated in the vector and recombinant vector is formed. Finally, recombinant vector is transferred in E. coli by the process of transformation. After transformation, transformed E. coli cells are selected on a medium which contain the antibiotic tetracycline. The transformed cells are ampicillin sensitive because it is inactivated by the insertion of insert DNA. Now these cells have tetracycline resistance but no ampicillin resistance. We select the transformed E. coli cells by insertional inactivation method. Now how we use insertional inactivation method for screening of transformed cell in this case. Now you see the non-recombinant vector, it contain both genes ampicillin and tetracycline gene functional. When we insert the DNA fragment for cloning in ampicillin gene, this gene become inactivated but tetracycline gene is intact. Thus non-recombinant vector contain both gene functional ampicillin and tetracycline, but recombinant vector contain only tetracycline resistant gene functional but the ampicillin resistant gene become non-functional. Now this ampicillin gene encode the enzyme beta-lactamase. This enzyme provide resistance against ampicillin antibiotic to the host bacterium. When E. coli contain non-recombinant vector, then that cell produce beta-lactamase enzyme, but when the E. coli cell contain recombinant vector, that E. coli cell will not produce beta-lactamase enzyme due to inactivation of the ampicillin gene. Now, what is the role of starch in selection of transformed E. coli cell? As we see in the previous slide, the ampicillin resistant gene encode an enzyme beta-lactamase. This enzyme convert penicillin antibiotic to penicilloic acid. This penicilloic acid binds iodine, and when iodine is in bound form, then it is not able to react with the starch and no blue color is formed. When we form a recombinant vector, then ampicillin gene is inactivated, so beta-lactamase enzyme is produced, and when there is no beta-lactamase enzyme, then no penicilloic acid is produced. When no penicilloic acid is produced, iodine is free and it can react with the starch and give blue color. In case of non-recombinant vector, ampicillin gene is active and it produces beta-lactamase enzyme which produce penicilloic acid which binds with iodine, and iodine is not able to react with starch and no blue color is produced. So the E. coli cell which contains recombinant vector give blue color when starch plate is flooded with iodine, and the E. coli cell which contain non-recombinant vector give no color when starch plate is flooded with iodine. In this way, when we do cloning in ampicillin resistant gene, transformed cells are selected on the medium containing soluble starch and tetracycline. After cloning, when E. coli cells are grown on this starch plate and after growth iodine is flooded, then transformed E. coli cell give blue color with iodine, and non-transformed cells doesn't give any blue color after iodine addition. Now what are the uses of pBR322? pBR322 is widely used as a cloning vector. When we have to amplify a DNA fragment, then we use pBR322 as a cloning vector. This vector is also used as a model system when we have to study prokaryotic transcription and translation, because one cloning gene is present in the promoter of the tetracycline gene. When we use this site for cloning of the DNA fragment, then this vector can be used as a model system for study of prokaryotic transcription and translation. This is all about pBR322 as a cloning vector. Thank you.